So let's get started. Hello, everyone. I'm Katja. I work at DeepScan Diagnostics. I will talk about liquid biopsies today. Let's get started. We have about an hour. I will not talk for that long. I'm usually a very quick talker. So there will be plenty of time for questions afterwards if you have any. I will give a brief overview of what liquid biopsies are, how we can use them, and then we will talk a little bit about cell-free DNA or circulating cell-free DNA and what it can tell about a dog's health, and also why we are comparing it to CRP and call it the next generation CRP. And then finally, I have some slides about how we can test cfDNA and how we can interpret those test results. So liquid biopsies. A liquid biopsy is basically just a lab test done on any bodily fluid. So it could be from blood, it could be from urine, saliva, other bodily fluids. And it is similar to tissue biopsy in a sense. So it is possible to use liquid biopsy to get some information about a disease type, especially the tissue type where the disease is located, sometimes about a specific cancer type, for example. It doesn't have as much of histopathological information as a tissue biopsy does. It's a fairly new technology. But the great things about liquid biopsies are they are minimally invasive. They are very fast, so you can get results within an hour in some cases. And most importantly, whereas if you think of a tissue biopsy, you already need to know where the disease is located, so where a tumor, for example, is located. And when you take the biopsy, it is hard to get all of the complexity of the tumor in that biopsy, whereas in liquid biopsy, you get everything because you take it from blood or you take it from urine or saliva or other liquid. It contains all the information from those tumors in the body. And they're both fairly affordable. I say fairly because it depends on the location and the type of test you're doing. But currently, I regard liquid biopsy to be complementary to tissue biopsies and also to imaging. But in future, it may well be that we will replace some of the current biopsies and imaging as diagnostic tools with liquid biopsies because they are so easy to take and easy to use. So what to test? There are a lot of different things and different levels as well. So when you think of a liquid biopsy, first if you need to think about what sort of liquid we're talking about. Most tests at the moment use blood just because the methods are most developed for blood samples. So there are well-functioning, characterized test kits for extracting DNA or RNA or proteins from blood, whereas urine, saliva, vials, other types of liquids, if you go to the manufacturers, they will say, yeah, we don't actually have a test kit available, and then you need to do a lot of R&D to develop those yourselves. So blood is usually the thing where we have started. We are also interested in other types of biofluids. And then again, with DNA, there's so much more known compared to RNA, proteins, metaboloids, for example, that it is the analyte of choice for us currently. But of course, there are others interested in many of these different analytes, and they all have their strengths. But then within the cfDNA or DNA, there are then multiple different things you can measure. You can look at mutations, you can look at copy number variations, gene fusions, insertions and deletions, different splicing variants that affect the gene function or the function of the protein. You can look at the gene activation or silencing patterns. We often talk about methylation patterns. So a lot of different things to look at. And like I said, we are interested in cell-free DNA or circulating free DNA. It's basically just DNA that is floating around in body fluids, let's say in blood, for example. Cells have released this DNA when they die, and the body quickly digests the DNA. So it's very fragmented. There is cell-free DNA or cfDNA in all of us. So it's normal to have it, because every time cells divide or they die, some DNA is released to circulation. But especially with growth, so in children and then during pregnancy, there is more of this cell division happening. So of course, more cell-free DNA. And then in some pathological processes, especially in cancer, but also in some systemic inflammation, for example, some autoimmune diseases. Of course, if there's trauma or surgery, a lot of DNA is released in the circulation as well. So then the levels will be elevated. But in healthy individuals, in adults, there is very little cell-free DNA in plasma, for example. And most of that comes from white blood cells, from the blood vessel walls, and from liver. And like I said, there are a lot of pathological conditions where more cell-free DNA is released in the circulation. Here's an example from cancer. What we know of cancer, when the cancer cells divide, this in itself can release some of the DNA in the circulation. Also, when the tumor growth is rapid enough, the surrounding tissue cannot nurture the tumor, and then it starts to die from the inside. So there's necrosis as well, and that releases a lot of DNA. And also there are these metastatic cells that are floating in the, or they are traveling in the circulation as well. So there are different ways of this DNA ending up in the circulation. And I just wanted to say at this point that when we talk about liquid biopsies, we talk about cell-free DNA, we talk about tests from others, from PetDx, from Volition, from CanCan, from us. These are not gene tests. So the purpose of all these tests is to determine whether there is disease in the body right now. So what is the current status? They are not trying to predict what the dog's future risk for disease is. So in that sense, our test, for example, it works as a next-gen CRP. And if you look here, there's an image I took from IDEX Catalyst CRP test brochure. Our test works in a very similar way. So, like I said, healthy dogs have very low levels of cfDNA. And then with the extent and severity of the pathological process, the level of cfDNA increases. The difference to CRP is there is a much larger variation. So the range of what is healthy and what is diseased, this is massive. So in CRP, you usually start to think about the disease between when there's more than 20 or 30 milligrams of CRP per liter. With cfDNA, you can already start suspecting this at a lower level, but then also at the upper end, you can have 10,000, you can have 35,000 milligrams per liter, which is quite a lot. And this usually only happens in recent major trauma, so a major surgery or with some blood cancers. So this is why we call this next-gen CRP. Because of the dynamic range, it gives more resolution. So we cannot say from a certain value that yes, this is definitely inflammation versus a benign tumor, but there is a bit of differentiation. So you would expect very low values with healthy dogs. Then if it's an infection, if it's local inflammation, you would have fairly low and stable values. With benign tumors, a bit more. With organ damage, for example, liver toxicity, you would expect a bit more. If there's trauma, even more, and then systemic cancer, and you will have, in the other end of the scale, you would have lymphomas, cystic sarcomas, maybe hemangiosarcomas, with a lot of cell-free DNA floating about. But that's not all. So also, because it is DNA, it is rather more informative than CRP in itself. So first of all, the way it is fragmented in the body, that already gives some indication as to what is the pathological process. So when there's cancer, these fragments tend to be shorter than when it's another pathological source. So it could be inflammation, autoimmune disease, or something else. So the fragment size distribution will be very skewed in cancer. Also, if we start looking at the contents of these fragments, we can then find out which tissue they come from, sometimes which disease they are related to. We can find mutations, we can find other variants that will give us additional information. So it's rather useful, but still I think these are complementary. So if we think of a CRP, it's one of the acute phase proteins produced by liver. And this is in response to stress. So the stress could be an infection, it could be inflammation or something else. But it's produced by liver, whereas cfDNA is just, it's fragmented DNA. It comes from all the dying cells anywhere in the body. And we and others have run some tests on or some comparisons of cfDNA and CRP. And the results so far are that CRP seems to be better at detecting infections, especially local infections. So ear infection, you may not see that much cfDNA in the circulation, for example. But cfDNA is better than CRP in detecting cancer, in detecting systemic inflammation and organ damage especially. And it also seems to correlate better with the disease severity and prognosis. So even if both are elevated, if cfDNA is very high, then it looks very bad for the dog. It means there's a lot of cell death happening in the body, even if CRP is not that elevated. We don't currently have enough information about autoimmune diseases, so we need to collect more samples and run these studies. But the point is that the combination of these two markers may really help us with the diagnostic accuracy. Then we can think about when to use liquid biopsies. There are lots of different options. We can use them. This is a case, a specific case from one of our former competitors. They were interested in cancer, and they proposed that liquid biopsies should be taken as serial tests for cancer screening. I do agree with that. And also a serial testing for treatment response. And again, I agree with that. Then there are other points where we don't currently have enough data to say how useful the cfDNA is, so aid in diagnostic targeted treatment selection, for example. But in general, I would say taking multiple samples a few weeks apart, or if this is for cancer monitoring or treatment monitoring, taking multiple samples just prior to each treatment, that's actually really helpful in looking at the dog's health and how the dog is responding to treatments, how the disease is responding to treatments, and whether there's relapse or disease recurrence. So here are a couple of test cases. We can use cell-free DNA as part of the regular health checks. And we have actually done this in Finland already. So we are collecting a baseline sample from dogs that are clinically healthy or appear healthy, and then we do regular sampling to check that the cell-free DNA level stays fairly stable. this could potentially be most useful in high-risk breeds to detect cancer and other diseases as early as possible, and in general in senior dogs. Also, if one already suspects a disease, like I said, cfDNA and CRP complement each other, so using both as part of a blood panel would be useful. And then the third case is to monitor treatment response. For this last case, we've run a collaboration in Finland and with our friends in the Netherlands and Slovenia. And so far, we have done this for 68 dogs. So the first sample has been taken in a treatment-naive patient when they arrive. We already know at that time which type of cancer they have. Mostly this has been either lymphomas or mast cell tumors or adenocarcinomas, but there have been a few cases of other types as well. So hemangiosarcoma, osteosarcoma, melanoma, soft tissue sarcoma, I think. Yes. And then we just follow how these how the treatment works. So like I said, we take the further samples just before each treatment. And the reason for this is the treatments themselves, because they kill cancer cells and cells in general, they will increase the cell-free DNA level very rapidly. So we take a sample just before each treatment to see how the dog has responded to the previous treatments. And then after the treatment regime has ended, we follow up the dogs for up to 12 months. And the results so far have been really promising. So as I said, cell-free DNA, it correlates really well with the extent and severity of disease. So we can see very sharp increases and decreases depending on how well the dogs respond to treatment. The most aggressive cancers, so I'm talking about lymphomas here because we have a lot of lymphoma samples, they often present a U-shaped curve. So the first sample, the baseline is very high because the dog hasn't treated yet. And then once the treatment starts, the cell-free DNA comes down very sharply for a while, and this can last for months. And then it starts increasing again, and sometimes this increase can be very sharp as well. It is a question mark still to me for how long we should continue testing and how often to get the best results. So there are some cases where we already are past the 12 months mark and we haven't seen any recurrence. Should we continue testing, or is this it? Has the dog been cured of cancer? I don't know. Maybe with some cancers, yes, but of course, I would like to continue these studies for longer. So here are two examples of lymphomas. On the left-hand side, a 6-year-old female Bernese mountain dog who initially had quite a lot of cell-free DNA, but then responded really well to CHOP treatment. And since then, all of the subsequent samples have shown very low levels of cell-free DNA. This level is already what we would expect in healthy dogs, so it's looking quite promising. Whereas the one on the right is quite typical to lymphomas if you have, especially if it's a more advanced lymphoma. So initially there's response to treatment, but then after a few weeks or maybe a month and a half, there's a turn to the worse, and eventually you'll end up having so much cell-free DNA in the blood and the dog becomes very poorly that a decision needs to be made whether to euthanize the dog or continue with palliative care. In contrast with mast cell tumors, mostly we have seen the curve on the left, or the diagram on the left. So you have the initial cell-free DNA, the baseline value, and it is usually not as high as it would be with lymphomas, for example, or hemangiosarcomas. But then there's a clear downward trend with treatment, and then also the value stays down. It doesn't come up. However, on the right-hand side, there is an example. This is an 11-year-old female Belgian Shepherd, where actually the dog already had surgery before the first sample was taken. So the dog was not treatment naive, but we wanted to follow up still to see how well the test works. And there was a clear downward trend. But after around 8 months, I think, we saw that the cell-free DNA was going sharply up. And then soon after, a decision was made to have another surgery because there was a recurrence of the mast cell tumor. And after that, we are back to very low levels. So this looks promising. If we look at all the samples, we have also looked at healthy dogs. I will next talk more about those. And with healthy dogs, if you look at the cell-free DNA values, as long as we control for pre-analytical factors, so how the samples are taken, how they are handled, how they are transported, how to extract the cell-free DNA, and so forth, the values are very stable. Whereas with cancer, you can see on the right, there's quite a lot of variation depending on the cancer and then on the time point. And this is again driven a bit by lymphomas because they tend to show this U-type curve. So we have also done health checks. We are currently running a 2-year follow-up study, and for this we recruited 400 healthy adult dogs in Finland. These are mostly dogs with higher than normal cancer risk. So we have Bernese Mountain Dogs, we have flat-coated retrievers, but also golden and Labrador retrievers, German and Belgian Shepherds, and Great Danes, Dobermans, and Staffies. And we take blood samples every 3 months, process and analyze those as normal. And if we find elevated cfDNA levels, these lead to further tests. So for most dogs, this is what we see. So here's a 7-year-old male flat-coated retriever and a 6-year-old female Great Dane. Very low levels of cfDNA, very stable levels as well. Here we have 2 dogs with cancer. On the left, a golden retriever, a 5-year-old golden retriever female that already had increased levels when we took the baseline sample. So outwardly, the dog was fine. So clinically, didn't seem to be suffering or nothing was found. But we were slightly worried that the first sample was so high. Took a second sample, but into a different tube. And this was lower. So we thought, oh, that's fine then. But then the third sample was again taken in the original tube type. So there's difference in the type of tubes people use as well. And this is very important. So we are currently recommending a certain tube type for everyone who work with us. So the third sample again showed elevated levels. And then we took another test a month after that. And that was very, very high level. And soon after that, the dog was diagnosed with renal adenocarcinoma and unfortunately had to be euthanized. But the lead time from the first test was 9 months. So if at that time we had already known that this is not normal for a dog of this age and breed, it would have given the dog a much better chance to overcome this disease. On the right, there's a 10-year-old female Belgian Shepherd, where the initial sample had very low level of cell-free DNA, looks completely normal. But then there's been a steady upwards curve. And after the third sample, the dog was then diagnosed with mammary adenocarcinoma and actually had surgery. the lead time between the second sample, where we said already that there's something wrong, and the surgery was 8 months. So it was almost a year from the first sample to the surgery. And then here's just a slide comparing us to Bolicious Nu.Q that's already on the market, and then the coming lymphoma test from IDEX. Just wanted to say that our test has been validated in more than 3,000 dogs so far, and more coming, of course, all the time. So we continue collecting samples from all over Europe, dogs with different cancers, with different chronic illnesses and inflammatory conditions. And we have created breed and age-specific reference ranges. We are the only ones who have done this, and this has really improved the disease detection rate for us. So I'm quite happy with these results. In summary, liquid biopsies are great. I think this is a tool that can revolutionize veterinary care of dogs. In human medicine, liquid biopsies are already used in cancer detection, in the treatment monitoring, in looking at minimal residual disease and so forth. They are quick, they are very affordable, they are very reliable. I think they complement well existing tools, especially CRP, tissue biopsies and imaging. And out of these different analytes, circulating cell-free DNA or cfDNA is the most promising, partly because the methodology in DNA side, it's more developed than with RNA or proteins. And there are different use cases, including health checks, especially for dogs with high risk or senior dogs, and as part of a diagnostic blood panel, and of course, treatment monitoring. This is how our test workflow looks currently. So it's very simple. A clinic takes 2 to 3 milliliters of EDTA blood, separates plasma, takes 1 milliliter of plasma into a fresh tube and freezes that, adds the dog's information into our data portal. So we like to know the dog's breed, age, sex, potentially neutering status, and also if there are any known diseases and medications the dog is already having, and then just ship the samples to us. We currently use DHL in Europe. That may change. And then our end, we extract DNA. We do some quality control. We analyze the sample and compare to the breed and age specific profiles to correct the values. And then we return those results to the clinic. And that's it. So just wanted to say thank you for listening. Contact these two guys, especially if you're interested to hear more. You can contact me directly if you're interested in the technology. Yeah, that's all I have. I'm going to stop recording now in case there are questions.